KBI Biopharma is a global contract development manufacturing organization that provides integrated development and manufacturing services to life sciences companies. Founded in 1996 with analytical sciences at its core, we've evolved into a trusted partner responsible for both mammalian and microbial development and manufacturing that, combined with our expertise in CMC, delivers robust clinical and commercial programs to our clients. We have approximately 1,700 employees spread across 6 sites globally, both in the Europe and United States, and we've serviced to date approximately 1,400 customers. Our cell line development capabilities are housed in our Geneva, Switzerland facilities. Cell line development then originates with taking that genomic DNA of interest for the protein that we want to express and stably transfecting our CHO-M cell line. We use the Sure technology platform to overexpress that protein of interest that indicated it required critical quality attributes at scale and in a volume great enough or titer high enough that we can then turn it into a molecule. To select the top clones, we go through and use automated cell culture techniques to identify top clones of interest. Those clones are banked as our research cell banks, and ultimately from those clones, we'll identify the top clone that is then our master cell bank that goes on to support GMP manufacturing. From there, then those research cell bank clones are transferred to our Venture Center facility in Durham, North Carolina in Research Triangle Park. Those top clones are screened both for upstream and downstream process development. So in upstream process development, what we're looking at is optimizing cell growth conditions, yield of our protein of interest, and understanding scaling up the process from 200 liters to 2,000 liters. On the downstream, then, we're focused on purification. So we want to purify, remove any impurities from our host cell, and purify our protein of interest while maintaining those critical quality attributes. We do this by working in parallel with our analytical development team, ensure that we're monitoring for host cell impurities, but also that we're monitoring other critical quality attributes. So this includes things like glycosylation, charge variance, and aggregation. So working in parallel in this way, we're able to narrow down those research cell banks and identify that one top clone from which we'll generate our master cell bank. This lays the foundation for the rest of the manufacturing program as we move ahead. So working in parallel with both process development and analytical development is our analytical and formulation sciences team. This team for our mammalian programs is based in Durham, North Carolina at our Hamlin Road facility. So this team performs the dual site qualifications in parallel with our analytical development team at Venture Center. Analytical Formulation Sciences is also responsible for carrying the molecule through phase 1 GMP manufacturing. So this includes in-process release and stability testing, alongside reference material generation and fit-for-purpose reference material characterization. As the program progresses and matures, AFS will continue to support the program as needed. This includes studies like force degradation, clinical and use and pharmacy manual studies, in-depth analytical characterization, and then ultimately, as you progress to late phase, implementation of specific residual HCP assays and targeted cell-based assays. Importantly, our analytical capabilities are really spread across all of our sites. What you'll notice is that both at our Venture Center in Research Triangle Park, we have strong analytical core there, led by our analytical development team that supports process development. We have analytical capabilities at our Hamlin Road facility in North Carolina, and we also have a deep analytical core characterization capabilities in our Louisville site in Colorado. They also have a particle core characterization facilities. It's important to mention that we are also capable, have a QC mass spectrometry lab and a dedicated cell-based assay lab in our Durham, North Carolina site to support all of the analytics testings that's required to bring a molecule to GMP. From there, once we demonstrate scale up of the process, so from going from 200 liters to 2,000 liters, we've performed our downstream development. We've integrated our analytical capabilities in line in real time, so we are ready to go as soon as that product is ready to advance to the GMP scale. At that phase, we're really then working with our clients specifically to draft what is their IND, and specifically the CMC portion of their IND, so the chemistry and manufacturing controls portion. This section of the IND filing comprises all of the effort that went into our cell line development, our process development, our analytical development, and any stability data that we have available at the time. From there, as the molecule progresses to later phase, we'll enter PPQ, product quality optimization, and then we'll validate not only the manufacturing process, but also the methods. Once we've entered PPQ and are headed to commercial, we then also work with our clients to file their biologics license application or their BLA. This application then builds on what we've already stated in the IND, but in addition to that, we will have the validated manufacturing process, our validated methods, and then prepare this dossier in partnership with our clients to get them through to FDA approval. This process is very complex and requires expertise at multiple phases, both in cell line development, process development, and analytical development. KBI is prepared and fully versed in offering that expertise to our clients when they show up from the onset, program initiation, day one. So KBI, being founded with analytical sciences at its core, is integral to who we are. We were founded in 1996 primarily as an analytics company. So long before we ever developed our integrated manufacturing services, we were an analytical company. This then has informed, we've worked with large pharma, medium-sized pharma, small startup companies, as well as even other CDMOs when they're looking for that deep analytical technical expertise. From the beginning, we have experience with specific mass spec-based assays, cell-based assays. We have in-depth experience with respect to method development. And as a result, as we transitioned into development and manufacturing, we're able to offer these integrated analytical services early on, right away from program inception. This has proved immensely valuable to our clients because it allows us to then troubleshoot and understand the challenges sooner, earlier at program inception, as opposed to waiting to the end to add on analytics. So we're actually integrating them each step of the process, all through process development, in method qualifications, and also in support of our analytical and formulation sciences activities, reference characterization, in-use studies, and all the rest. Importantly, this means that we are well-versed and experienced in a host of assays. This includes, size exclusion aggregation measured by size exclusion and capillary electrophoresis, glycosylation patterns, understanding charge variation, a host of these assays, understanding impurities that we are well-versed and experienced with and understand how to apply the methods for a quality product at the output. Towards this end, KBI utilizes our Sure technology expression platform. In addition, Solexis has put out our TranspoEase system, which uses a transposase enzyme-based targeted insertion of the DNA of interest for our protein of interest at optimal locations within the genomic DNA. This is advantageous when combined in conjunction with our CHO-M cell line, which has a very short doubling time of 15 to 17 hours. So we take that process of cell line selection very seriously, and specifically the process of the clone that we're going to carry forward from our research cell bank to our master cell bank. And to do this, then we want to understand not only the critical quality attributes of the protein that's being produced, but also our yield, our titer, and we carefully measure and monitor how many generations for that cell line is stable for. So to do this, we performed our next generation sequencing to understand if there are any unwanted variants in there. In addition, we are able to demonstrate fewer than 5% of unwanted variants. So the decision about whether to go and use mammalian versus microbial expression is really dependent on the molecule of interest. Many of the biologics that we manufacture and are putting forth for the clinic require glycosylation to actually have that immune engagement within the body. That requires that we use mammalian expression. But for some smaller proteins, smaller molecules, or enzymes that maybe don't require post-translational modifications, we might recommend microbial expression. So microbial expression itself is a faster timeline, has a lower cost of goods, and is very efficient in its production. KBI does have a microbial center of excellence located in our Boulder, Colorado site. And this site is unique in that development, manufacturing, both clinical and commercial, are all housed under one roof. And in this way, we're able to move very quickly through the process without delays. KBI also has a proprietary E. coli cell line called Pure coli, which has been genetically engineered. So about 20% of the genome has been modified so that we're reducing expression of native proteins that we don't need. So in this way, our engineered Pure coli cell line is more efficient and yields a higher yields at the end of that product. And similar to our mammalian systems, you can expect expertise and understanding all the way through cell line development, process development, analytical development, and clinical and commercial GMP manufacturing and release. We're able to maintain everything there within that system. In addition, it's important to point out that while KBI offers expertise end-to-end across all of the various stages of bringing a product to market, really, we also offer a number of standalone services. So these standalone services can be maybe you just need cell line development, or maybe you just need a method developed for a specific assay, or maybe you're late phase and are looking for a cell-based assay that you can implement to support your product. We have what's called the STAT, the KBI standalone portal. And in interfacing with this portal, then you can log in, have an account, and you can request different services and needs that you have. Importantly, to understand through this central request hub called STAT, we also have other online interfaces and tools to communicate with our integrated clients. Towards this end, we have Program View, which is our proprietary software that offers in real time as you can get, both raw data and decision-rated data for our clients, so they can see as we're progressing through process development and testing the analytics to understand those really critical early phase, really milestones and decision points. They're able to log in, interface with us directly as that data is available, and make key decisions to move the program forward as fast as possible. So that's called Program View, and we offer that to all of our clients. If you have any additional questions or information, please feel free to contact us through our website at kbibiopharma.com. We would be more than pleased to work with you, learn about your needs, and share with you our capabilities. We're also absolutely open to hosting site visits. Come and get to know us, and we would love to support your program all the way through CLD to manufacturing.